This animation is going to show us the Lac Operon, and we typically see operons in prokaryotic cells. Operons help these cells regulate gene expression. Typically, RNA polymerase will bind to their promoter and transcribe the genes to make the enzyme lactase. The enzyme lactase breaks down the sugar lactose. The yellow structure in this animation is known as lactase, and it's a protein that's going to break down that lactose, which is a disaccharide. It breaks lactose into the monosaccharides glucose and galactose. RNA polymerase will also make something called the repressor from the regulator gene. This repressor is another protein that's going to bind to the operator. Whenever the repressor has binded to the operator, RNA polymerase will not be able to transcribe the genes to make lactase. You might ask, why would a cell want to stop gene expression or protein production? This is because if there's no lactose around, there's no need to make lactase. So just as a review, the whole function of the repressor is to stop transcription, which will stop translation of the protein lactase. Because again, there's no lactose around, why waste material and make lactase? However, whenever there is lactose around, it will bind to the repressor, which will change its shape. Once the shape of the repressor has been changed, it won't be able to bind to the operator anymore, which will allow RNA polymerase to make and transcribe the genes to make lactase. Because now that there is lactose around, there is a need to make lactase. So this mRNA will go to make the enzyme lactase to break down the lactose. Once the enzyme lactase is produced, it will go and break down all the lactose in the cell. After the lactose is broken down from the repressor, it will go and bind to the operator again, stopping transcription. But this won't happen until the lactose is broken down by the lactase. Again, this whole process just regulates gene expression.